Hi, Grandma. 2 weeks ago, we talked about innate immunity, the one that we have at birth. Today, we're going to talk about the second type that we acquire throughout our life, and it's called adaptive or acquired immunity. It's often classified into two types, divided into two types: humoral immunity and cell-mediated immunity. So, what is cell-mediated immunity? It is an immune response that involves T cells and antigen presenting cells. So it's basically cells mediating with each other. T cells are one type of lymphocytes that are formed in our bone marrow. They migrate into our thymus where they mature. Then they leave they leave the thymus and move to reside in lymph nodes where they become mature and naive T cells. Based on their service markers, they are classified into either CD4 plus or CD8 plus. On the other hand, antigen presenting cells are cells capable of ingesting, processing, and presenting antigens or the signal or the activation signals to these T cells. Examples of antigen presenting cells include macrophages, dendritic cells, and B cells. Now, let's talk briefly about the sequence of events in cell mediated immunity. Let's assume that a microorganism gain access to one of our tissues or organs. It will be recognized and identified by one of our antigen presenting cells, APCs, for example, dendritic cells. These dendritic cells will ingest these invading microorganisms and move toward the nearest station, which are lymph nodes. Here's what lymph nodes looks like. You can see here the dendritic cells, which carrying the invading microorganism, are presenting them to the naive T cells, and based on the nature of this invading microorganism, CD4 plus or CD8 plus uh, T cells can get involved. And here where the cell mediated immune response will be initiated inside the lymph node. Under the influence of interleukin 2, these activated T cells will differentiate into one of its categories, which is T helper 1, T helper 2, cytotoxic T cells, or CD8 plus memory cells. Uh, as I said, under the influence of interleukin 2, this differentiation will be followed by clonal expansion of that particular type, and each of these types has its own function. For example, T helper 1 will secrete interferon gamma and tumor necrosis factor, and these are active against intracellular microorganism. T helper 2 will secrete interleukin 4, which stimulate B cell production of antibodies, especially IgE, and it will also secrete interleukin 5, which promotes the development of eosinophils. You can see here T helper follicular T helper cells will secrete interleukin 21, and that will stimulate stimulate high rate of antibody production by B cells. T helper 17 will secrete interleukin 17 and interleukin 22, which are active against gut and lung microorganisms. Cytotoxic cells attack virally infected cells or tumor cells through the secretion of specialized pore-forming protein called performine that allow the intracellular injection of a series of proteases that will facilitate the killing of those virally infected cells or tumor cells. Lastly, I want to mention an example of cell-mediated immunity, tuberculin skin test, which involve injection of killed TB particles into our skin. That's a kind of delayed delayed type hypersensitivity reaction, which involves the activation of helper T cells. And that's what I have today, man. Grandma, see ya.